We are now exiting the review portion of this lecture, and we're going to start to enter into new territory. Molecular interactions occur when two or more molecules associate with one another. In biology or physiology, we often see these molecular interactions forming new protein complexes, forming, certainly it's necessary for substrates or so molecules to interact with enzymes if they're going to be catalyzed, if the enzyme's going to catalyze a chemical reaction. And so these molecular interactions, they're facilitated by a number of different properties, including solubility, whether the molecules are hydrophobic or hydrophilic. And when we're dealing with large molecules, we usually have domains that are lipophilic versus hydrophilic. We've got the ionic charges, we've got polar charges, we've got the three-dimensional shape. All of these end up dictating which molecules will interact with each other and which ones will not. Molecular interactions occur between a variety of different molecules. For example, two or more peptides or proteins can interact with one another. This is actually how we get those quaternary structures. We also will get interactions from cell to cell through these proteins that interact with one another. Proteins can interact with carbohydrates, proteins can interact with lipids, proteins can interact with DNA or RNA, and then quite commonly proteins will interact with a small molecule. And those small molecules are often used in chemical reactions, or they're transported or moved based on their binding to the protein. DNA binds to RNA, RNA binds to RNA, lipids bind to lipids, and so on. This is not an inclusive list. Here's an example of a protein that is interacting with DNA. And so we actually see, this is actually just a small segment. So you notice we see the alpha helix here. And it's, and here we actually see the disulfide bridges. That's what you're looking at right here is a disulfide bridge. And here too. And here too, they often stabilize double helixes, and in this case what they're stabilizing is, for some reason it's called a zipper. And I guess that kind of makes sense because it looks like this is one side of the zipper and this is another side of the zipper, and this zipper interacts with the DNA molecule. We're going to talk a little bit later on in a different lecture about why certain proteins interact and how they affect transcription. But this is likely a regulatory protein that's going to affect the transcription of DNA. I've talked already about the molecular shape and how they fold, so I don't want to revisit this. This essentially looks at that molecular shape. But I do want to focus on this as we start to look at the shape. What ends up happening, here's a larger protein. You can actually see some, again, the alpha helixes here. We've got a few beta sheets here. But what I really want you to focus on is these red and these blue sections here. And what those are little pockets. They're small. But when we're dealing with a large molecule and its ability to interact with, say, a small molecule, these smaller molecules tend to tuck themselves away inside those pockets. And that allows this protein sometimes to modify those molecules, or vice versa. The molecules can actually modify the shape, the three-dimensional shape of the protein. These molecules, one of the most important things that regulate whether they interact with the protein is hydrophobicity. So a small hydrophobic molecule will bind into a pocket that contains hydrophobic amino acids. Vice versa, hydrophilic molecules will tuck themselves into a pocket that contains hydrophilic amino acids, either the polar or the ionic. And shape, of course, and other chemical properties determine that interaction as well. Now, here's some vocabulary for you. The pockets, these little hydrophobic pockets, it depends on what type of molecule we're dealing with. If we're dealing with, let's say, a transporter, or a carrier protein like hemoglobin, which carries oxygen in the blood. The job of those proteins is not to catalyze or change, not to catalyze a reaction. They don't change the molecule that binds it. Hemoglobin doesn't change the structure of oxygen, for example. And so when we have a protein like that, we call the protein is typically referred to as the target molecule or the target protein. I have that on another slide coming up. And then the pocket itself is referred to as a binding pocket or a binding site, or sometimes a docking site. Those three terms are used very commonly interchangeably. Now, if this protein is instead an enzyme, and enzymes are going to catalyze a chemical reaction, and that means it's going to change the molecule that binds. So the small molecule that binds will be altered in some way, some chemical reaction that occurs. If that's the case, then we actually call that binding pocket an active site, because it's in that active site that the enzyme can catalyze the reaction. Now, what about the little molecule? Well, ligands are the term that we use just generically for any small molecule that docks or binds with a larger molecule that is not chemically changed. On the other hand, if we're dealing with enzymes, we call it a substrate or a reactant, or sometimes even a reagent. Substrate being the most common, because in this case the substrate is changed. If you think about in terms of art, let's imagine you're a painter and you have this blank canvas, and now you're going to put paint on the canvas. You've changed the canvas by applying the paint. And so we use substrate in the same way in regards to the enzyme. Or, think of a ball of clay as you manipulate and change shape. We're changing the substrate. And so that's why we use the term substrate, as it kind of brings up that mental image there.